So we're going to talk about postsynaptic potentials, or PSPs. These are graded. What that means is that if you increase the stimulus size, you increase the amplitude of the PSP. That's in contrast to the action potential, which the stimulus is coded in the frequency. So if you increase the stimulus, you get an increase in frequency of action potentials, not an increase in the size of the action potential. And that's why we call action potentials all or nothing. Now, there are two general types of PSPs. There's excitatory PSPs, which are EPSPs. These increase the chance of firing an action potential. IPSPs are inhibitory postsynaptic potentials. These decrease the chance of firing an action potential. So a simple way to think about it would be, if the stimulus is a depolarizing stimulus that's graded, that would be an example of an EPSP. And if the stimulus is causing hyperpolarization, the membrane potential is becoming more negative, that would be an example of an IPSP. Now, overall, when a neuron is getting inputs from different cells that are releasing different neurotransmitters, there could be some that are EPSPs, there could be some that are IPSPs. And really what overall the cell is going to do is going to be dependent on the summation of all of those inputs. So you have to add them all up, and you could end up with a net depolarization, or you could end up with a net hyperpolarization. It sort of depends on what the different stimuli are. Now, I want to use an example of a very specific EPSP that is named after the cell type that it is found on. And this is going to be the skeletal muscle end plate. So I want to go and show us an example here of a lower motor neuron, which is what we name the motor neuron that releases acetylcholine onto the skeletal muscle end plate. By the way, these neurons are large diameter, heavily myelinated neurons of the A alpha type. And the acetylcholine that's released here, you can see that it's coming from a vesicle that's fused, released into this junction, which we call the neuromuscular junction, because it's the junction between a motor neuron here and the skeletal muscle. Now, acetylcholine has receptors that it can bind to, and in this case, you can see these green receptors here are an example of a ligand-gated ion channel. So this is a receptor that is an ion channel. And not only is it an ion channel, but it's a mixed cation channel. What that means is that when the channel opens, and that's going to be acetylcholine binding here, there's a electrochemical gradient for sodium and potassium. When the channel opens, the pore is large enough to actually let sodium and potassium go across. So we know that we have an electrochemical gradient favoring sodium. We have a high concentration of sodium on the outside relative to the inside. We have a high concentration of potassium on the inside relative to the outside, and the membrane potential at rest is a negative number. So when the channel opens, sodium is going to go in through the channel and potassium is going to go out. Now, you might think that these just cancel each other out because I have a cation going in and I have a cation going out. Well, the magnitude of these currents is different. The size of the sodium current is larger going through this channel than the potassium current. And the main reason why, as you can imagine, is that the driving force for sodium is larger at rest because of the negative charge that's in the cell and at the membrane potential. There's further distance from the equilibrium potential of sodium and the membrane potential at rest. And so when the channel opens up, that is going to result in a larger current. Because the potassium equilibrium potential is fairly close to the resting potential, you don't get as large of a current due to the fact that the driving force is small. Now, when you get a net depolarization here, so overall, the inward sodium current, which is going to be a negative current, is going to be greater than the outward potassium current. Overall, then you're going to get a depolarization of the membrane, and this is going to be in a graded fashion. It's going to be based off of how much acetylcholine is being released and how much of these nicotinic receptors are being activated. And so this is a graded depolarization. This is graded, and that is what we call an end plate potential, or EPP. So this is just a specific type of EPSP. It's because we're on the end plate of the skeletal muscle, we call it an EPP. And if this size of the EPP is large enough, remember, you're going to open up voltage-gated sodium channels that are found on the skeletal muscle. They're present there too. Remember, these are voltage-gated. They activate in response to depolarization. So you're actually going to get sodium current going through these sodium channels. If we get enough of them open to get to threshold of the skeletal muscle, we could get a muscle action potential. So if large enough, we can get what we call a muscle action potential. It's specifically a muscle action potential because we're on skeletal muscle, not a neuron, which we just call those action potentials. I'll also point out that we are talking about an action potential still on skeletal muscle, and what's not depicted here are voltage-gated potassium channels, which have to be present here because that's how you're going to get repolarization of the skeletal muscle membrane to allow you to deactivate those sodium channels and then fire another muscle action potential if enough acetylcholine comes around again. And so this is how the motor neuron here is releasing acetylcholine, causing the skeletal muscle to generate electrical activity, which ultimately results, when you get these muscle action potentials, in skeletal muscle contraction through the process of excitation contraction coupling.